Welcome to yet another BIOS E1A tutorial. In this tutorial we'll be going over problem 8 on practice problems 2-2. If you haven't gone through the practice problems yet, please pause this video and take a minute to review them. Also in this video I'll give a brief overview of meiosis that will help with solving the problem, but if you don't have a good handle on the small details of prophase, metaphase, anaphase, and telophase yet, it might benefit you to go through those lecture slides again or to rewatch that part of the lecture video. Question 8 tells you the following. Aneuploid gametes are cells that contain the wrong number of chromosomes. Aneuploidy occurs as a result of non-disjunction during meiosis. And then it asks, at what stages of meiosis would non-disjunction occur? Briefly explain. And then part B, imagine a cell had a diploid chromosome number of 4. Create a diagram to illustrate the effects of non-disjunction of one pair of homologous chromosomes in meiosis 1 versus meiosis 2. So right away from the question, we can see that there's a dead giveaway that non-disjunction can happen in both meiosis 1 and meiosis 2. So how does this happen? That is, how can a cell divide into gametes that have an incorrect number of chromosomes? This is a single cell that is about to undergo meiosis. It's diploid with 2n equal to 2. So it has one chromosome from each parent, one red and one blue. As you can see in the diagram, the cells have already been duplicated, and we have one set of duplicated homologous chromosomes. During meiosis, this cell is going to divide to form 4 gametes, each of which is haploid with n equal to 1, as we can see in the products at the bottom of this slide. So the question is, how do we go from a single diploid cell, DNA replicated, to 4 haploid gametes? This is accomplished through 2 divisions. One division of the original cell, and another division of each of the resultant daughter cells. We have here a single diploid cell with a duplicated genome. During meiosis 1, the 2 homologous chromosomes line up along the metaphase plate, and each is dragged to an opposite pole by the spindle fibers. The cell then divides the cytoplasm, and we have 2 daughter cells, each with one of the homologous chromosomes. You see those daughter cells here. Note that this cell has only one set of homologous chromosomes, but the same process happens in cells with a greater number of chromosomes. Each pair of homologs pairs up with its partner, and one moves to one pole and the other to the opposite pole. This is what we call the reduction division. It reduces the number of chromosomes in the daughter cells to half that of the parent cell, and this is a very important point. Meiosis 1 is a reduction division. The parent in this case has 2 chromosomes, and the daughters have only 1. So meiosis 2 then takes place to account for the fact that the resultant daughter cells still have a duplicated genome. In meiosis 2, each chromosome lines up along the metaphase plate, now without its homolog to join because they've been separated into different cells. Each duplicate chromosome then has one of the sister chromatids pulled to each of the poles of the cell, such that each resultant daughter cell gets only one copy of each chromosome. We can see the products here, each with the same number of chromosomes as the meiosis 1 product, but only a single copy. And this way, this is an equational division. So we can say that whereas meiosis 1 was a reduction division, the number of chromosomes was cut in half, meiosis 2 is an equational division. All of the gametes have the same number of chromosomes that the meiosis 1 products had. In short, meiosis 1 cuts the number of chromosomes in half by separating homologous chromosomes, and meiosis 2 then makes sure that each gamete only has one copy of these chromosomes. So what can go wrong for gametes to end up with an abnormal number of chromosomes, as described in the question? One way this could happen is with an error in meiosis 1. Perhaps an error occurs and meiosis 1 turns out not to be a reduction division, as it should be. That's exactly what happens with non-disjunction. We can see here that this cell underwent meiosis 1, but an error occurred and both chromosomes segregated to a single cell, rather than one to each daughter cell. The division is not a reduction in chromosome number for both product cells. Instead, one product receives both homologous chromosomes and the other none. This is non-disjunction in meiosis 1. But as the question asks, which stage of meiosis 1 does this occur in? In prophase 1, the cell has 2 duplicated condensed chromosomes. In metaphase 1, the chromosomes pair up with their homologs and line up along the metaphase plate, as shown here. What should happen next, in anaphase 1, is that one homolog separates to either end of the cell, as the arrows indicate here. What happens in non-disjunction is that both duplicated chromosomes go to the same pole. When the cells separate during telophase and cytokinesis, we get one meiosis I product with no chromosomes, and one with a duplication of that chromosome that failed to segregate properly. So we can see that non-disjunction can happen during anaphase of meiosis I. From there, everything will proceed the same. The chromosomes will line up along the metaphase plate in meiosis II, and sister chromatids will separate to create 2 daughter cells for a total of 4 gametes. 2 of the gametes, as you can see here, will be deficient for the chromosome that failed to segregate properly, and the other 2 will have a duplicate. But what happens if everything goes properly in meiosis I? The homologs line up and separate during anaphase of meiosis I, and the reduction division happens as usual. At this point, it's possible for sister chromatids to fail to properly segregate during meiosis II. This is non-disjunction during meiosis II. We can see on the right that sister chromatids went through proper separation, and the equational division produced 2 haploid gametes. On the left, however, sister chromatids failed to separate, and we're left with one cell with duplicate copies of a chromosome, and one cell with no copies of that chromosome, meaning essentially that the division is not equational as it should be. This is our second type of non-disjunction during meiosis II. So we can answer part A of question 8 now. We saw that non-disjunction can occur during both meiosis I and meiosis II. In both divisions it occurs during anaphase, as this is when the chromosomes and sister chromatids are actually separating. It's this failure to separate properly that creates meiotic products with an abnormal number of chromosomes. Now that we have seen this and understand how exactly non-disjunction can happen, part B of this question is trivial. It reads, imagine a cell has a diploid chromosome number of 4. Create a diagram to illustrate the effect of non-disjunction of one pair of homologous chromosomes in meiosis I versus meiosis II. All we have to do is apply what we've just learned. This diagram represents a diploid cell with 4 chromosomes. There are 2 sets of homologous chromosomes, 2 from the mother and 2 from the father. Each parent contributed 1 short chromosome and 1 long chromosome. Which chromosome came from which parent doesn't really matter in this case, and they've been color-coded just to keep track of them. The chromosomes have already been duplicated, as coded by their thickness. A non-duplicated chromosome, in this case, would be thinner, just for the sake of this diagram. The diagram is showing the chromosomes of metaphase I of meiosis. Homologous chromosomes have paired up along the metaphase plate, crossing over has occurred, and they're preparing to separate during anaphase I. Anaphase I happens without any problem, and the 2 meiosis I products have a reduced number of chromosomes, as they should, 1 long chromosome and 1 short each. The chromosomes line up along the metaphase plate and prepare to separate during anaphase II, and non-disjunction occurs now. The sister chromatids comprising the long chromosome on the left failed to separate into 2 cells, and both ended up going to the leftmost product. This leaves us with a duplicate of that chromosome in one gamete and a deficiency in the other. This provides us a diagram for non-disjunction during meiosis II, answering part of half of part B, rather. Now let's diagram non-disjunction during meiosis I. In meiosis I, homologous chromosomes should separate. In non-disjunction, there's a failure for this separation to happen. So one meiosis I product ends up with both homologous chromosomes for any given pair that undergoes non-disjunction. In this diagram, the long homologs failed to separate during meiosis I, and the cell on the left ends up with both of the long chromosomes. These meiosis I products then undergo normal meiosis II, and sister chromatids separate to produce 4 gametes. The gametes on the left are diploid for the long chromosome, and those on the right are deficient in it. These are the products that result from meiosis I non-disjunction. So to review the practice problem, we found that non-disjunction occurs both during anaphase I and anaphase II, as this is where the chromosomes are actually separating. Non-disjunction is simply a failure for them to separate as they should. So in meiosis I, it's a failure for the homologous chromosomes to separate, and in anaphase II, it's a failure of the sister chromatids to separate. In the latter half of the video, we produced 2 diagrams demonstrating this process for both meiosis I and meiosis II. Thanks for watching, and I hope this video helped with your understanding.